understand the pancreatic hormones that secreted from pancreas and what's their role in regulate metabolism. We're also going to discuss the insulin structure and how it was synthesized. Again, the most important physiological role of insulin, we're going to discuss this as well. And finally, after this lecture, you should be able to describe all those important factors that regulate glucose homeostasis. So pancreas, from the location here, you can see pancreas is actually has endocrine section and an exocrine function as well. So on GI physiology, we are going to discuss the exocrine portion of the pancreas. This lecture will only focus on the endocrine portion. So you see the highlights in the circle, that's the endocrine portion. We call this also islet of Langerhans because all the cells they are like clusters, like an islet. So we call this portion also, we call the endocrine portion of pancreas, we also call it islet of Langerhans. Now you can see in the middle of the picture, that's the pink, the light color, in the middle, that's the islet of Langerhans. So the islet of Langerhans are surrounded by the exocrine portion. On the right hand of the picture, you can see the islet of Langerhans actually consists of 3 different cell types. They are alpha cells, thus consist of 20%, and the majority is beta cell. And around 10% of the cells are actually delta cell. So these 3 different cells, they secrete different hormones. Beta cell is secrete insulin, and alpha cell secrete glucagon, and the rest of the 10% of delta cell, they produce somatostatin. I'm not sure if you still remember somatostatin. We discussed this in the hypothalamus section. That's one of the hormones that's secreted from hypothalamus. Now we are discussing this one in the pancreatic section because this hormone is also secreted from the delta cells in the pancreas. Okay, let's look at how insulin was synthesized and what the structure looks like. So insulin is actually a polypeptide hormone. It's a long chain, a straight line. It composed of alpha chain and beta chain. That's the final structure of insulin. But the production of insulin actually went through several steps. So they start with proinsulin, and in the endoplasmic reticulum, they produce the preinsulin. And once they were released from the endoplasmic reticulum, they are matured. That's the function of insulin. So proinsulin actually has 4 different sections. They have an N-terminal, like you can see, that's the brown color, and in the blue section, the blue line, the blue color and the pink color, those are the major part of insulin. So that's beta chain and alpha chain. So it's the C-peptide. You see the green long picture there, right? So that section is actually C-peptide. So the proinsulin consists those parts. And once this proinsulin was produced, they moved to the endoplasmic reticulum. In the endoplasmic reticulum, the N-terminal was cut down. They got rid of that, that's useless. And then the portion left was the A chain and the or sometimes you call it alpha chain or beta chain. And that was still connected. So the A chain and beta chain was still connected by the C peptide. But that structure is not stable anymore. So that disulfide bond was added in the endoplasmic reticulum. Now the insulin is ready to be released. Okay, before it's released, they actually cut down, right? They cut the C peptide, and the remaining part is alpha chain and the beta chain that they are combined by the disulfide bond. Okay, so the only insulin, the alpha chain, beta chain, that's action, that's the physiological active part, and the C peptide is not has no physiological function anymore, but the C peptide has a much longer half-life, so it's stable in the blood. However, insulin is only stay in the blood for around 6 minutes, and it's breaking down. So that means because insulin and the C peptide, they are produced in the equal amount. So we can use C peptide to assess and monitor the endogenous beta cell function, right? So sometimes when you want to detect the beta cell function and to see whether the insulin produced, is it from endogenous or is it from exogenous? So we use to detect and monitor the C peptide level. Okay, so that's insulin was synthesized and what the structure looks like. Now, what's the physiological role of insulin? Everyone knows that insulin reduces the glucose level, but how? Okay, so first thing we need to understand the target organs. So the major target organs in the human body is liver, skeletal muscle, and adipose tissue. And how does it perform its role? So after each meal, right? After each meal, the glucose level is elevated. So the glucose is floating around the bloodstream. It won't hurt. So it has to go somewhere because they can't stay in the plasma, in the blood. Now, because glucose was floating around, they move to the pancreas. They give the signal to pancreas beta cell. And once the pancreatic beta cell gets the signal, insulin is released. So once insulin released, they bring glucose. Okay, they bring glucose into liver cells, skeletal muscle cell, and adipose tissue. So now that way the plasma glucose level is maintained in the normal range. Even no matter how much glucose, no matter how much nutrients you eat, but the glucose level is stable. Okay, so that's the role of insulin. Now, because glucose is like a small molecule, right? They can stay inside the cell, but that's not perfect way to store them. So that's why glycogen is the best way to store glucose. Okay, so another function of insulin is they produce. Okay, once the glucose is inside the cell, they produce they produce glycogen. Okay, but this only occurs in the liver and skeletal muscle. How about fatty acid? So after we eat, after each meal, the fatty acid level and triglyceride level is also elevated, right? We don't want that to happen. They have to go to somewhere. So insulin, again, perform the same role. They bring free fatty acid into the adipocyte, and inside the cell, the free fatty acid and triglyceride is bring back into the adipocyte, and there they produce more fat tissue and store there. Okay, so add up together, the physiological role of insulin is really very important. Again, let's repeat, let's just summarize. They are working on liver, skeletal muscle, and adipose tissues. They increase the uptake of glucose. Okay, not only glucose, they also increase the uptake of amino acid. Second role, they increase the glycogen production. Okay, in the liver, the muscle. The third, they stimulate lipid synthesis from fatty acid and triglyceride. The very last one, we didn't mention in the previous slides, insulin also stimulate potassium uptake by cells. Okay, so that means they also push the potassium back into the cells. In that way, the potassium level in the blood is also maintained in a very narrow range because insulin is going to bring the potassium, okay, back into the cells. So it's also play is the video. I won't be playing here. Now let's discuss what's regulate insulin release. So from this one, you can see after each meal, right? For example, after breakfast, after lunch, after dinner, after half an hour, around takes about 20 to 30 minutes, there's going to be a peak of insulin. Okay, after lunch, another peak. After dinner, the third peak. So the major stimulation of insulin secretion is elevated blood glucose level. Okay, so that's the major stimulus of insulin secretion. So again, once the blood glucose, once the insulin causes uptake of glucose into tissues, blood glucose level drop. So insulin level declines when the blood glucose level declined. Okay. So now let's look further about how glucose can stimulate insulin secretion. Let's look at this in a cellular level. Okay, the reason I bring this slide is because there is a medication actually works on the potassium channel, okay, that stimulate glucose induced insulin secretion. That's the sulfonylurea drugs. So glucose, right? So after the glucose moved to the pancreatic cell, they were induced, okay, through GLUT2 receptor, they were uptake, okay, into the beta cell. In the beta cell, they produce ATP. Once ATP production was introduced, ATP production was elevated, they closed the potassium channel. Once the potassium channel was closed, that leads to depolarization of the pancreatic beta cell membrane. Once the depolarization happened, the calcium channel is opened. Once the calcium channel is opened, insulin is released back into the plasma. Okay, there are several drugs that works on GLUT2. There's a medication works on the potassium channel as well. So those medications can be used to stimulate glucose-induced insulin secretion. Finally, let's discuss about the glucose homeostasis. So we discussed about glucose stimulate insulin secretion and insulin secretion. Elevation of insulin level brings the glucose level back to normal. So what other factors that can induce glucose, can stimulate glucose level? Okay. So first, the glucocorticoid. So the cortisol can elevate the glucose level. Okay. And that's one hormone can elevate glucose level. Another one, epinephrine and no epinephrine can also elevate glucose level. The third one is growth hormone. Remember, when we talk about growth hormone, because growth hormone can elevate glucose level. That's remember in people who has acromegaly, they have the gluconeogenesis capacity. They may have insulin resistance, they may have diabetic, right? So these three factors, they can stimulate, they can elevate plasma glucose level, either through glucose neogenesis, can through the glycogen breaking down through different mechanisms. The end result is to increase blood glucose level. Okay, so these three hormones, you must memorize it. Those are the three stimulating factors that participate in glucose homeostasis. Okay, so together with glucagon is another very important hormone that increases blood glucose level. So add up together, there was four very important factors that can increase blood glucose level. They are glucocorticoid, epinephrine, norepinephrine, growth hormone, and glucagon. Now let's look at the major action. Let's combine everything together and look at the major action of insulin and look at what's its final effect on the different levels, such as on the glucose level, amino acid level, and the fatty acid levels. Okay, so let's look at the left-hand column, right? So they decrease everything, right? When the insulin was released, they reduce blood glucose level. They reduce blood amino acid level. They reduce blood fatty acid and ketone acid level. They also reduce potassium level. You must memorize those four different effects, right? So all nutrients we eat is either glucose or fatty acid or amino acid. Insulin decrease all these three factors. Okay, so that's the function. So why does they produce this effect? Again, this table summarizes everything. Okay, because glucose can lead to the uptake. The insulin can lead to glucose uptake into the cell, right? Through insertion of glucose transporter. Insulin can lead to the glycogen formation in muscle and liver. And in the meantime, gluconeogenesis decrease and the glycogen lysis decrease. Okay, gluconeogenesis means the blood can be produced through amino acid and fatty acid, right? And the glycogen lysis means the breaking down of glycogen. So 1, 2, 3, through this four effect, the final outcome is to reduce blood glucose level. So what happens to the amino acid? Because insulin can stimulate amino acid uptake, they also increase protein synthesis in the tissue. Okay, they inhibit protein degradation. So the final outcome of insulin is to reduce blood amino acid level. Same thing in the adipose tissue. Insulin increase the fat deposition, decrease lipolysis. The lipolysis, the breaking down of fatty acid is reduced. That's happened in adipose tissue. And in the liver, insulin inhibit keto acid formation. Okay, so that's the major role. The reason patient has a DKA is because insulin levels low. The inhibition of keto acid formation was reduced. We're going to discuss this when we discuss the pathophysiology of DKA. So again, insulin induced the uptake of potassium into cell. So the final outcome is decrease the potassium level. So those are the major action of insulin and its final effect. Those are very important. After you understand the action of insulin and its effect on glucose on the blood level, it's going to be easier for you to understand the pathophysiology of diabetic ketoacidosis and other diabetic related complications.